So Thomas, thank you for introduction. First, I want to say that it's a big pleasure to me to be here today, and I'm going to talk about the main subject of my research, which is basically high-resolution 3D mapping of the human hypothalamus towards a comprehensive cytoarchitectonic atlas. But before going into details of the project, I just want to briefly remind you a few general information regarding the hypothalamus. So basically, in the ventral part of the brain, you see this two roundish structure, which is mammillary body, which is nuclei of the thalamus, and they define the most posterior part of the hypothalamus, which is mammillary zone, and we defined also two other zones. One is tubular zone, and most anterior, it's a preoptic zone. Functionally, I would say the main function of the hypothalamus is to maintain the homeostasis of our bodily processes. But how hypothalamus achieves this? First, by controlling the autonomic nervous system, which is, so it regulates vital functions such as heart rate, blood pressure, respiration, water balance in our body. It also involved in the endocrine system because it controls the production of the hormones from the pituitary gland, which is in turn influences the growth, reproduction, and metabolism processes. Last but not least, the hypothalamus play a key role in the limbic system, meaning this emotions, memory, and different type of behavior, such as aggressive behavior, for example. So advancements in the spatial resolution of the modern neuroimaging, enabling the study of the structure-functional relationship within the hypothalamus and the living human brain. But as you see on the right side of the screen, it's difficult to localize the brain activation from the functionally different nuclei, which is not visible in the routine MRI. So the problem is there is no detailed histological references of the hypothalamus microanatomy in 3D space, at least till the end of this presentation. Yeah, and moreover, speaking of the clinical relevance, there are a bunch of diseases which happened because of the disturbances in the hypothalamus. The most relevant one, it's a cluster headache, sleep disorder, symptoms in multiple sclerosis, thermoregulation, panic disorder, aggressive behavior problems, and eating disorder. So, and in the recent publications, there were like studies on applying the deep brain stimulation to deal with the severe cases of specifically cluster headaches and eating disorder. And as we know, the success of the procedure is highly dependent on the precision of localization of the electrodes in the brain. That's why our goals of the study actually was to provide the detailed set of architectonic maps of the hypothalamus in BigBrain as a 3D reconstruction. Then it's to generate probabilistic set of architectonic maps which basically quantify the intersubject variability, which cannot be obtained solely from the BigBrain. Last but not least, we also want to quantify the cellular architecture of the nuclei and evaluate the difference in site architecture between them. So speaking of the workflow, we took the 10 brains, 5 came from female, 5 from male body donors, and among these brains were BigBrain 1 and 2. Then we obtained the undistorted MRI volumes of the brains to later on, based on this, reconstruct the original volume of the brain. Then we sectioned the brain for 20 micrometer thickness slices, stained them for cell bodies. Then I performed the cytoarchitectonic mapping in every 15th digitized section with a 1 micrometer resolution based on the cytoarchitectonic criteria, which is mainly like the type of neurons, the size, distribution, and density. When I finished in all 10 brains, the delineating, we based on the previously obtained undistorted MRI, as I said, we get the 3D reconstructed volume of each brains and then normalize them and transfer to commonly used reference spaces, MNI, for example, column 27, and then we overlap the regions with each other and get the probability maps. For example, here is a dorsomedial nucleus, and this color-coded polygon basically depicts the probability of finding this particular nucleus in this position in this voxel, with the probability like 100% in the red color in the middle, while if we go more in the edge, you see this is like a rather green or even violet color, meaning that the probability is rather 50% or 10%, or we can find this nuclei just in 5 or 1 brain. There is a few other examples, like from ventromedial nucleus from the same area and the arcuate nucleus. Overall, there were like 23 nuclei, and we wanted to produce also the maximum probability map, where we combine all the probabilistic map together. And for example, here, you see this in the reference brain, we assign each voxel to those of the nucleus which has the higher probability and get this maximum probability map of the hypothalamus. So, as I said, we worked with the 1 micrometer resolution, and we really want to benefit from it and go deeper to study the microstructure of each of the nuclei. And for this, we used, for example, the contour proposal network, which was developed by Eric Utschulte, our colleague from both Düsseldorf and Jülich. And he yesterday gave us a great talk and explained this network in the more details. So I just briefly remind you that with the contour proposal network, basically it allows us to label each pixel in the microscopic image as this histological section with an index which represents the distinct cell, which in turn allows us to perform the reliable and precise cell identification. Here in this histosection, I zoom in 2 particular places. The first one is, here, for example, it's a border between the posterior hypothalamic area and ventromedial nucleus, as well as tubermammillary nucleus. And here, for example, you see that how the cytoarchitectonic features are differ between the nuclei, like the size of the neurons, their density are quite differ. So, and the method itself identify each neurons in this nuclei, and we were able also to upload our delineations as a mask to extract the specific characteristic information from each of the nuclei to study the cell density, cell morphology, shape analysis, and spatial distribution from each of the nuclei. The second method was the texture analysis, which was developed by Dr. Sebastian Bludau from Jülich. So we also analyzed more than 6,700 regions of interest from all 10 brains with 1 micrometer resolution. We extract 21 modified texture features, such as contrast, homogeneity, and others, to quantify the cellular architecture of the tissue. Then we reduced these 21 features to 4 main components using principal component analysis. And then we also assessed the difference between nuclei by applying the Kruskal-Wallis test. So to visualize the results, I prepared this heat map, where on the right side you see the bar, which represents the degree of differences in the texture features, starting with the 0 green color, meaning that the texture features are quite similar between nuclei, going up to 4, a red color, meaning that we obtain the significant difference in each of the 4 components that we obtained from the principal component analysis. Since the table are rather big, so I forgot to say, on the X and Y axis there are all the structures that we identified in the hypothalamus. So since the table is rather big, we just, as an example, have a closer look to this particular square where we can compare 4 nuclei. And let's start with this, comparing anterior periventricular nucleus with supraoptic nucleus. So according to the method, we obtained the high difference in the texture features, but you may ask how it looks in the section. So on the right, we see that in the anterior periventricular nucleus, there are rather like sparsely dense nuclei, smaller paracellular nucleus neurons, while in the supraoptic nucleus, they consisted of the bigger histaminergic neurons with a high density. But if we compare this uncinate and intermediate nuclei, which basically have a rather smaller difference in the texture features, and indeed in the histosection you see the size of the neurons are quite similar and the distribution are also quite comparable. As I said, we also wanted to create the 3D reconstruction of the hypothalamus in the BigBrain dataset. For this purpose, I delineated each 10 section in the BigBrain dataset, and then we used the deep learning based tool which was created by Dr. Christian Schiffer from Jülich, and we upload my delineations to Atlas UI software, where these manually delineated sections became manually delineated training sections. So meaning that we trained the model with these sections, and the model itself helps us to predict the delineations in all the sections in between previously manually delineated sections, resulting in more than 400 annotated sections with 1 micrometer resolution. And we were able to perform the 3D reconstruction of the hypothalamus, and there's a model on the right side. So with this 3D reconstruction in the BigBrain, we basically capture the shape and extent of each nuclei of the hypothalamus with the cellular precision, and we also revealed the topographical relationship between the nuclei. So the model itself, it's rather complex, so I suggest we just have a closer look on one hemisphere. And as I said, we defined 3 main zones in the hypothalamus. The most posterior, it's a mammillary zone, in the middle, a tubular zone, and the preoptic zone, most anterior one. So in the mammillary zone, we identified 8 nuclei, as well as some wet matter structures which goes there, and you immediately see that the shape are quite differ, but what is more important even, like the volume are quite different. See this, the one of the biggest nuclei in the hypothalamus, lateral hypothalamic area, which has a volume of more than 250 cubic millimeters, comparing with this one, lateral mammillary nucleus, for example, one of the smallest one, it's just around 1 cubic millimeter. Yeah. So if we go further to the tubular zone in the middle, so this zone is particularly involved in the production of the hormones, namely paraventricular nucleus and supraoptic nucleus are produced the oxytocin and antidiuretic hormone in our body, while the arcuate nucleus are mainly produced the releasing factors which controls the production of hormones from the pituitary gland. So in the most anterior zone, it's preoptic zone. Here I think it's most densely occupied by the smallest nuclei, which is intermediate nucleus, and uncinate nucleus, which is just about half of cubic millimeters. So our map basically captured the shape and extent of all 23 hypothalamic nuclei, which breach the microscopical level of organization of the hypothalamus with the macro level, which provided by the MR imaging. The map can be used as anatomically plausible parameters for brain modeling and simulation, while the probabilistic cytoarchitectonic maps quantify the intersubject variability and show the localization and extent of the area in the 3D space. And the maps could be used as a tool for interpreting and comparing neuroimaging data and may contribute to better understanding of the functional organization of the hypothalamus and its nuclei. With this, I'm gonna end my presentation and really thank you for your attention. Thank my supervisor, Professor Katrin Amunds, as well as my colleagues from Düsseldorf and Jülich, namely Eric, Christian, Hartmut, and Sebastian, for your help and contribution throughout the project, which is still going on. So thank you.